Hello, my name is Minju Kim, and I'll be presenting on the modulation of angiogenic pathways in AGS cells, the role of SU14813 in VEGF-driven tube formation. To briefly introduce myself, I am a 9th grader currently attending Seoul International School, and here is an overview of the research. So to move on to some research motivation, gastric cancer ranks 3rd among cancers in terms of mortality, and the survival rate for patients with metastatic gastric cancer remains low. Current treatments include surgery and chemotherapy, but their effectiveness in advanced stages is limited. Angiogenesis in cancers like gastric cancer is essential for cancer cell survival and growth. In particular, VEGF plays a key role in promoting angiogenesis. Therefore, drugs that inhibit VEGF activity can play an important role in cancer treatment. This study therefore aims to determine how effectively SU14813 inhibits VEGF-induced angiogenesis and explore its potential as a new therapeutic option for gastric cancer. So the goal of the study was to analyze the effect of SU14813 on VEGF-induced angiogenesis, cell mobility, and cancer cell viability. To do this, I designed 3 main experiments. But before we move on to the experiments, first I cultured AGS gastric cancer cells using DME medium. The cells were grown to about 70 to 80% confluence and used in experiments after approximately 48 hours. To analyze cell viability, the first experiment was a WST assay. Cells were treated with VEGF and SU14813 either separately or in combination and were cultured for 24 to 72 hours. WST region was then added and absorbance was measured to assess cell survival. To evaluate angiogenic tube formation, I used Geltrex, a substitute for matrigil and a useful material for observing angiogenesis. AGS cells were cultured on top of Geltrex and treated with VEGF and SU14813. The degree of tube formation was then observed. Next, we conducted the cell migration analysis using a wound healing assay. Cells were seated in 12-volt plates and a linear scratch was made using a pipette tip. After treating with VEGF and SU14813, the wound closure was observed over 24 to 48 hours. There were mainly 4 experimental groups. First is a control group, second is a VEGF-treated group, and third and fourth are the SU14813 treated groups with different concentrations. This allowed comparison across a range of conditions. So the phenotype analysis was conducted using a microscope, and we also conducted a statistical analysis, and the results were expressed as mean plus minus standard error, and statistical significance was tested using t-test and ANOVA. Now moving on to the results. For cell viability, VEGF significantly increased the viability of AGS cells, while SU14813 reduced viability in a dose-dependent manner. With 2 nanomole, viability decreased by about 14%, and with 50 nanomole, it decreased for about 45%. When the 2 substances were used together, viability dropped by nearly 50% compared to VEGF alone. Over time, VEGF led to a consistent increase in viability compared to control, while SU14813 treated groups showed inhibited cell growth. And in conclusion, SU14813 effectively suppressed VEGF-induced cell survival effects. Now moving on to some phenotype analysis, in the VEGF-only group, the tubes were longer and were more complex. In the 2 nanomole group, the tubes were thinner, and in the 50 nanomole group, tube formation was nearly absent. The number of tubes also showed dramatic differences. In the VEGF-treated group, there was almost double the amount of increase in tube count. In 2 nanomole, there was almost 50% decrease, and in 50 nanomole, very few tubes were formed. It was interesting to see that tube thickness also decreased and VEGF-induced structures collapsed under SU14813 treatment. Finally, I conducted the cell migration analysis, and the results were that VEGF enhanced cell migration and promoted wound healing compared to the control. SU14813 treated groups showed significantly reduced wound healing, and in high concentration group, not only did the wound not close, but the area even widened, indicating a strong inhibition of migration and proliferation. In conclusion, the study confirmed that SU14813 can inhibit VEGF-induced angiogenesis and cell migration. This suggests that inhibitors like SU14813 could be potential therapeutic agents in gastric cancer treatment. The limitations of the study lies in the fact that the research focused mainly on AGS cells. We need to test whether similar effects occur in other cancer cell lines as well. So in future work, I would like to investigate other angiogenesis inhibiting anti-cancer drugs that are already developed or under development. By addressing these limitations, I hope to make meaningful contributions to the pharmaceutical industry through cancer drug development. That concludes my presentation. Thank you so much.